Yes, I think it's just a little quiet. We had a loudspeaker last time. There we go. Hello, welcome to another installation of Chemistry Senior Seminar Series 2022, Week 9. We are going to be joined by Chris today, and Stefan Nevert is going to be introducing him.
Okay, so again, as always, it is my distinct pleasure to introduce our speaker today. Chris Hoang came to us from Vietnam. He's been working in my lab. He worked in my lab for some time. I met Chris his first term at Lawrence, where he demanded that I let him into organic, and I said no, I don't like to let freshmen in. And he's like, but I love it, and I was like, everybody says they love it because they want to get it. Chris loves organic chemistry. As a freshman, he sat in on my advanced organic chemistry class pretty much every day, right, before I let him into organic. And then I said, okay, fine. And he excelled in those classes. And he's really carried a love of building molecules throughout his time here. And so he'll talk a little bit about the work that he's done with me. But mostly he's going to talk today about kind of what total synthesis can do. So what we can contribute to the world around us, and kind of the role that synthetic chemists can play in making the world better, and how that really blends a sense of social responsibility with the kind of cool, puzzly challenge that really kind of excites both Chris and I. So again, we've worked on tempering that excitement for this talk a little bit, but I'm sure he will talk at length about any questions that you might have after it. So without further ado, I'll give you Chris Hoang.
All right. Thank you, Stefan. Hi, everyone. My name is Chris, and Stefan have introduced me. So today I'm going to talk about total synthesis as a resource in drug discovery. So on the right side of the screen here, we have three kind of wonky molecules with a lot of rings. Artemisinin, one thing like MMV0202505 with a lot of numbers, and the last one here with a bunch of question marks. So these three molecules here are going to be the star of my talk today. So let's get started. All right. So first, let's talk about a little bit about history of malaria. So back in the medieval, back into the 1400s, 1600s, even 1700s, 1800s, there was a disease that always spread around the humid areas surrounding marshes and swamps, and people thought about it. People thought about it was like from the miasma around it. So the Italian, they came up with the disease name combining the two Italian words, mal, which is mean bad, and aria, which is air, basically bad air. So malaria was thought to be caused by bad air. But not until like 1880s, there's a French scientist named Charles Louis Alphonse Laveran. Sorry for my bad pronunciation. I don't speak French. So he discovered that parasite was the main cause of malaria. So let's fast forward 200 years to the modern day. If nowadays malaria is still a problem, it is well known that malaria is a mosquito-borne disease and is just caused by parasite. People with malaria often experience fever, chills, and flu-like illness. If it's left untreated, they may develop like severe complications and even lethal. So according to CDC data back in 2019, there are over 200 million cases worldwide, and over 400,000 of them end up dying, and it is bad. So in the US, around 2,000 cases are diagnosed every year. In 2010, World Health Organization has published a kind of guideline about using artemisinin-based therapies as a treatment for malaria. So, okay, I just bring up the artemisinin word. Let's talk about the history of artemisinin. So here on the right side, this is Artemisia annua. It's a plant. And interestingly, it is a very famous traditional Chinese medicine. It has been using like thousands and thousands of years across the history of China. Artemisia, back in the day, so in around like 1940, 1950, when malaria was like kind of bad around the world, it was kind of endemic around the world, scientists back then tried to find a way to treat malaria. There was like chloroquine back then, but then malaria developed the chloroquine resistant. So they drive to find a new drug to treat malaria. So they start screening plants and plants. Artemisia was one of the plants they tried to screen, and I mean, like the extraction from Artemisia was one of the they tried to screen. They extract the plant with hot water and ethanol, and however, the extraction gave some result, but it's inconsistent. Sometimes it worked miraculously. Sometimes it does not at all. So scientists were like, maybe something we did wrong. So after that, one scientist in China, she discovered that if she tried to extract these plants with ethyl ether, but in very, very low temperature, like not that very low temperature, but low temperature, not boiling, and then wash the extraction with alkaline solution like we did in our lab with the separating funnel to get rid of all the acidic byproducts, we would get some neutral extraction. And that extraction turned out to be really effective against malaria. That active component in the extraction were later on isolated in 1972 and named artemisinin after the plant Artemisia annua here on the right side of the screen. And that discovered would then, in 2015, awarded the Nobel Prize in Physiology and Medicine to Yo-Yo for the discovery of artemisinin. So it was much later on. About artemisinin. So this is the parasite. So this is a cell of the parasite here. There are many things going on the screen, but let's focus on some small thing. So artemisinin, when it gets into the parasite cell, it do three major things. First of all, it inhibits detoxification pathway here. It blocks the polymerization from heme to hemozoin, which is eventually build up the heme concentration inside the parasite cell and toxificate the cell, parasite, which eventually kill the parasite. The second way it can interact with the parasite cell is to promote the alkylation of proteins, which kill the cell. And last way is to promote the depolarization of membrane, so basically break down the parasite cell. So it seems artemisinin is really good. However, we do face some problem with artemisinin. We call it artemisinin shortage. Why? Because artemisinin can only be found from the leaf of the plant, or the Artemisia plant. You cannot get artemisinin from the root, you cannot get it from the stem, only from a tiny fraction of the plant, the leaf only. The extraction gives really low yield. So even the most recent research on the extraction of artemisinin from the plant in 2018 by CIFC, the yield only about 0.71% of the mass give out the artemisinin. Okay, we have a low yield. Can we just mass planting the tree, mass planting it? Well, it is kind of hard. It cannot be mass planting. Why? Because it requires fairly long growing period, about 200 days. It requires really strict growing environment. So it requires average temperature to be 10 to 15 C, which is way lower to the average of tropical area. However, it requires a really high rainfall, annual rainfall, more than 1,000 millimeters per year. And it requires a really specific soil condition. It requires light soil with a deep topsoil. So it brings us to the need that we need an efficient way to mass produce artemisinin. So chemists, we love to build molecules. We can extract the molecule from nature, it's a good way, but can we just build a whole big molecule from the ground up? That is where total synthesis come in. So total synthesis is about synthesize a complex molecule from really simple, small, commercially available precursors. And the great thing about total synthesis, the chemist has the total freedom to modify the molecular structure according to their desire. Of course, we still have to follow the law of nature, we cannot give carbon with 5 bonds. So yeah, all right. We have the potential to mass producing by, sometimes we can just scaling it up by 10 times, 100 times. Maybe, let's hope that scaling up will work. All right, so total synthesis of artemisinin. Artemisinin is a great molecule. So the first total synthesis was back in 1983. It consists of 7 steps with 9% yield. 9% doesn't seem a lot, but 9% is more than 10 times about like you just extract purely from the plant. So it's great. It's like 10 times better than getting straight from the tree. However, we can see here, the starting material is still fairly complicated and it still takes like 7 steps. So as a synthetic chemist all around the world, we have access to many different tools. We can create bonds, we can break bonds, we can make rings, we can break rings. But the toolbox is a mystery box to us chemists. We have to find the best way, the optimal way that we can create the molecule in the fastest way, but also give the highest yield. So research going on in 2012, there are a new research how to make artemisinin. And now we have this unsaturated cyclohexanone, like fairly simple molecule. And within only 5 steps, the research group has successfully total synthesized artemisinin with 29% yield. So the yield has been like 3 times increased comparing to the first total synthesis back in 1983, and the steps has also been reduced from 7 to 5. But total synthesis, 5 steps, seems awesome, right? Well, not quite. So total synthesis is also really hard. Look at, this is the total synthesis. This is everything that research group in 2012 was about. Only 5 steps, so 5 arrows here. 1, 2, 3, 4, 5, 5 arrows. But we have a lot is going on here. We have zinc, we have copper, we have lithium, we have aluminum, we have palladium, we have molybdenum. What are those doing with organic chemistry? Well, to synthetic chemistry, sometimes we simplify these. We can look at these as name reactions. Name reactions are well-known reactions around the world, have been proven to be working, and extremely powerful tool. So look at that. Let's see, the very first step here from 4 to 7 here in the paper, well, it's actually just conjugate addition and alkylation. From 7 to 5b here, we have the Shapiro reaction. 5b to 9, we have a hetero-dialysouter. So the word hetero here is basically like, instead of just raw, pure carbon making a ring, you have non-carbon going in and making a ring. So next is here, from 9 to 10, we have the Wacker reaction, using a palladium to make a ketone here, which is pretty cool. And the very last step here with molybdenum, well, the molybdenum there is to generate the singlet oxygen inside the pod. So we can cyclize into artemisinin. So basically, all that actually work there is a singlet oxygen. All right, so with anything, we live in a world that always evolve. Human evolve, animals evolve, also bacteria and parasite evolve. They will eventually build up a resistance to the drug. So back in the earlier my talk, I talked about the research on artemisinin was originally driven by the chloroquine-resistant malaria. So we have chloroquine-resistant. We also have artemisinin-resistant malaria nowadays. And in fact, it's a real specific mutation, the PF-CALH13 mutation in the P. falciparum parasite. And that mutation gives the parasite the resistance against artemisinin. Why do we have to worry about that? Well, because that one mutation has become predominant in Southeast Asia, namely Cambodia, Myanmar, and surrounding countries. And eventually, if that keeps spreading, it's going to be dominant species around the world. And we no longer have a good, effective treatment against malaria. So now it is in need for a new antimalarial drug. We take inspiration from nature. Nature is cool, but sometimes we can inspire ourselves. We can build big machines and screening and testing molecules that doesn't exist in the nature, but look like a drug molecule. That's when a foundation called Medicine for Malaria Venture came in. So they published a collection of 200 drug-like structures, and the structure was selected based on the result of phenotypic screening against that parasite, P. falciparum, which causes malaria. They selected compounds based on the few criteria, such as it has to be commercially available, so we can do more research on those. it has to be maximized in the structural diversity. So synthetic chemists can tinker with the molecule and make it to our desire. And most importantly, we are trying to fight against the malaria. So it has to show some potency against the malaria. And here, this molecule here, like MMV020505, this molecule shows some potency against malaria. But what about the structure? Why this molecule is so special? So, well, the reason is you can divide this molecule into three very distinct parts: a hydrophobic head, a linker, and a basic chain. So if you divide that into three parts, can we build the molecule part by part? Well, yes, that is when retrosynthesis came in. So imagine if we break the carbon-nitrogen bond here, this bond, we can use a fairly effective reaction called reductive amination to build that C-N bond, quick and easy. And the C-C bond between the hydrophobic head and the linker, we can use another reaction called the Meerwein arylation. Why these reactions are so miraculous? Well, because if we just switching out the starting material, for example, here we have the R2 groups instead of the dimethylamine here. If we switch out that R2 groups with anything like piperidine ring, we would have a different compound. Or here in the compound number 4, if this arylamine will switch out, if the R1 will switch out with different thing, such as here we have fluorine and chlorine. If we switch out as bromine and chlorine, now we have a different compound. So because of having that modular structure, by just switching out the starting material, we can mass produce analogs of the same molecule, and then we can start testing those molecules to see which one has the highest potency against malaria. But also, switching out molecules, switching out atoms, also change the structural of the molecule. And what's so big deal about the structural activity relationship? So there's a thing called the partition coefficient that is described how a solute distributed between two immiscible solvents. So in medicinal chemistry, we usually use octanol and water because octanol is the one that can mimic the environment inside the cell. So we can have a rough idea how the drug will dissolve in the cell once we intake the cell. And how we can calculate that partition coefficient? Well, this is our trustworthy separating funnel. We add an equal part of octanol and water, and of course, our solutes in there. So the partition coefficient is going to be the concentration of drug in octanol phase, which is the top layer, divided by the concentration of drug in the aqueous phase, which is the bottom layer. And that number is usually reported under the term log of P. So log of P is controlled by molecular structure because if we switching out molecules, it's going to be a different thing, and it's changed the solubility of the molecule. Log P can be predicted with computer program. Computer program is a really powerful tool. So if we just chunk in different molecules and calculate and try to simulate to find which molecule has the most optimal log P for the drug intake, we can then try to bring that kind of molecule to the lab and synthesize it in real life. So I did some calculation with the program called Chemicalize to generalize the impact of different ring substituents make on this molecule. So here, if the baseline I used was using R groups here with hydrogen, it has a log P of 2.267. So whenever I change the R group here into one of these substituents, the log P of the molecule will change by the delta of log P. So for example, if I change the R groups into the OH instead of H, my log P now turn into 1.767 instead of 2.267. So it is a really powerful tool when we can aim for a particular log P and try to push other ring substituents. It helps us to choose which ring substituents to use in the synthesis. So That's the one side of the equation. We also have the amine, the side of the amine. We can swap out different amines. So if the core line is just like NH2 here, the log P will be 2.935. For the R is like H, which is NH2. We change that into like long basic amine chain. It will increase the log P. Well, however, if we switch that out to somewhat a salt, it will vastly decrease the log P, which increase the water solubility of the molecule. So after that, after we have some idea about like which kind of fragments we choose, which substituents we can choose, which kind of basic amines we can choose, let's go a bit about the synthesis of the molecule that I want to make. So it is an analog starting from 4-fluoroaniline, and the reaction is simple. It's just 4-fluoroaniline in a Meerwein relation, and then I can create a CC bond between the arylamine and the furan, the furfural base, and I can do the reductive amination between the furfural and the amine here, giving me the final product. So the question is, how can I find out if I can actually make the final product? The good thing is we have access to a tool called nuclear magnetic resonance. So this is the NMR spectra of my molecule. When I look at the NMR spectra, well, there's a lot of peaks. Let's assign those first. We have from A to I. So each of these peaks represent the hydrogen on the molecules. Some of those hydrogens are really easy to be picked off, and some of those might be a little bit trickier. So on my molecule, the easiest hydrogen peak I can pick off should be those two. Why? Because there are no neighboring molecule around those two hydrogens. So it should show up as a singlet on my NMR spectra. So for this one, I would expect an integration of 2 under a gigantic singlet. And here, because we have 2 methyl groups, I would expect integration of 6, but also a singlet. So where are they? Those are this E and H, which represent to that and that hydrogen. So what's next? Well, the next easiest thing to pick up on the NMR spectra, usually if it is the furthest to the right hand or furthest to the left hand. So it means like if it's furthest to the right hand, it is the most shielded. And if the furthest to the left hand is the most de-shielded. So let's go and look at these 2 peaks. Those are fairly de-shielded. All right, let's zoom in. So the reason why it's splitting weirdly here is because there's a fluorine on this atom. The fluorine interferes with the NMR spectrum. Without the fluorine here, the A and B peaks would be, I would expect to be look at like a clean, nice doublet of doublet. And here we have C and D left over. Let's zoom it in. We have 2 doublets at C and D, each with the integration of 1. Those should be here, the C and D. There's nice and clean doublet because they only have 1 neighboring molecule, 1 neighboring hydrogen. So D neighbor with C and vice versa, C with D. All right, what do we have left? We do have F and G left. So F and G, they are both really nice looking triplet. Triplet means it should have 2 neighboring molecules. So it should either be this under this carbon or this carbon because the only one CH2 is the CH2 at the middle here. We can assign F and G like that based on the de-shielding. However, if we have a certain method such as HMBC, hetero multiple nuclear resonance, correlation, we can have a correlation between the peak E here, between these peaks and that peak, so that if F and E show up under the HMBC, we can correctly assign that this one is F. And if these 2 show up with a G, we can assure that we assign G correctly. All right, so that is one of the molecule that I made. Oh, we also have one last thing that I forgot to assign, but like, hey, it's a peak I. That's the only thing that left. It should be that one. However, when we zoom it in, it is a bit weird. We would expect I to have 1, 2, 3, 4, 4 molecules, 4 neighboring hydrogens. So it should be a quintet. Yes, a quintet. However, because it is right, we have a large Y peak. I would assume that would be the water to interfere with that. So the integration value for this peak is incorrect. If I can write out the sample and retake the NMR, I would expect really nice and clean quintet with an integration value of 2 representing for 2 hydrogens. All right. Yes, it would look something like this. Okay, so after that, after we have synthesized a few with different basic amine chain or a few with different ring substituents on the hydrophobic head, we can start thinking about what are the different linker we can use. So we can make 1,3,4 oxadiazole, and so instead of the 2 carbon here, now I swap out with the 2 nitrogens. We can also swap out with the 1,2,4 triazoles. So instead of the oxygen here, now it is all nitrogens on the ring. These molecules are all interesting and promising. Maybe they can show some potency against the malaria. So I did start the talk with artemisinin. I did talk about the molecule that I try to make. And I always dream of, like, let's combine what I did with what I dream of, like one gigantic, big, complicated total synthesis. So I want to combine the artemisinin into that scaffold structure. So I want it to be a furan-based with 3 modules, a hydrophobic head, a linker, and a basic amine chain. And it has to have the endo peroxide of the artemisinin, which theorized to be one of the main reasons why artemisinin is so effective against malaria. So there we go. The good old question mark, question mark, question mark from the beginning of the talk. It appears. It has 3 parts: amine chain, a linker, which is a furan, and an endo peroxide. So the question is, how can I make this molecule? It is just big, big questions. So let's assume that somehow I can get to this one. Well, let's think a bit about how can I get to this one. I can get to 1 from 2 using the Heck reaction. From 3, I can get to 2 using thionyl chloride, using lithium triterbutoxyl aluminum hydride, and end up using like reductive amination, like I just said, just talk using with the other molecule to make that amines nice and good looking. So here, these 2 hydrogen, we have like dihydrofuran base. I can make that using the Birch reduction from the furan with bromo here, and the furoic acid. This is the furoic acid, but the carboxylic acid here will direct the Birch reduction to push that 2 hydrogens into the position that I want. 4, so I'm at 4. I would think, how can I make a 4? Well, 4, we can start with 5, which is fairly simple molecule. We can cook that in acid and treat it with Jones reagent, and then terbutylithium and carbon dioxide to push that carboxylic acid group as a last step. So okay, we have 1. We want to make that ring. So the paper in 2012 did react there. The last step they did react with a singlet oxygen. Can I also react that with singlet oxygen? So this is a reaction with a singlet oxygen. Well, it will have, first of all, have a 2+4 cyclization here, like DL's outer-like mechanism into that. And then the lone pair of the oxygen will come out and push the new CO bond away, forming a peroxide like that. And now this O minus is going to be really reactive. It's going to bend over and react with a carbonyl center and push that up. Okay, we now at this part, we have a new ring here with the 5-member rings with double bond O represent the peroxide. We have the O negative charge. If everything going accordingly. This O will bend over to the C carbon number 2 here, doing a 1-2 addition and push the electron up here like this to the oxygen and cyclize nicely to make a 7-member rings. However, we have to also compete for another 1,4 reaction here because we have that alkene here. What if that O bend under there, going into carbon number 4, the electron will push up and then eventually go into the oxygen. It will also cyclize into these two products. This is a 1-2 reaction, will give us our question mark, and then 1,4 will get us to a surprisingly cool-looking molecules, but not what we intended to do. Yes, of course, it still have a ring here. There's endoperoxide, there's a furan, there's an amine chain, but can this also have some reaction, some effect against malaria? Or, so I come up with another question. Well, can this kind of reaction, can I choose one of these molecules as a dominant product? Can I drive it using thermodynamic versus kinetic? So I hop onto ChemCompute and try to calculate the electronic energy of these. And the electronic energy of these two are really close to each other. So it seems that they can both exist, assuming that all the reactions I've talked about so far actually work. All right, so yeah, it is kind of good question to think about, like how can I direct it toward the 1-2, 1-4 in the future research. Let's talk a bit about the 1-2 product because that is the first goal when I started to talk and I wanted to think about that 1-2 product. So the one thing special, the most special thing about that 1-2 product is it has higher oxidation state comparing to artemisinin bicyclic system. By I mean the bicyclic system is those two here. And why does it has higher oxidation state? Well, look at the artemisinin. It is saturated. Yes, it's really, it has really high oxidation state because it has like a bunch of oxygen in the ring. However, here we have a double bond, which open countless of opportunity because like double bond can react with stuff. And yes, that one. So, and second thing, it has the modular scaffold structure. So imagine if we can actually substitute that double bond with new R1 and R2 here, we open a lot of possibility of tinkering with this molecule because yes, it still has the hydrophobic head, which we can swap out R1 and R2. We have a linker, we have the amine chain, which we can swap out the R3 here. All right, so as a conclusion, total synthesis has a gigantic important role in drug discovery. We can use total synthesis to mimic the nature, to recreate what nature is good at, or we can use total synthesis tinkering a molecule to our need. The structure here, this molecule here, MMV020505, is really suitable for modification and optimization. As we have seen, we can just tinkering with the fluorine and chlorine or ring substituents in general. We can tinker with the amine chain, and we can swap out the center ring. The artemisinin-inspired MMV derivatives may have some beneficial characteristic like, well, it has endoperoxide, maybe it can react. It has that double bond, open up many, many possibilities for synthetic chemistry. And the synthetic pathway and mechanism of artemisinin-inspired MMV derivative can still be improved because, well, we can shorten it, we can make it more economy, and we can maybe do a lot of things about that. So that's it for my talk. I would like to thank everyone that have helped me until this day. I would want to thank my colleague at the Summer Research Experience, my Chem 680 colleagues, Dr. Rachel Dowdy, and I would say thank especially to Dr. Stephen Deppert, who has helped me since the day one I got here to Lawrence. And yes, this is the citation, and any questions?
All right, thank you, Chris. He's ready to take your questions. if you have them. Allison.
So when you did the chem compute calculations, you said the energy that you had. Do you go back to that slide?
Yes. All right. Can compute. This one.
Yeah. So can you tell me more about the energies that you calculated using chem compute?
Oh, yes.
It's pretty big.
Yes, it is pretty big because there are 1, 2, 3 ring system here in the molecule up here, and there's a lot of oxygen, so it's in a really high oxidative state. Also, what I did with the chem compute, so basically what I did is I just used the chem compute function to optimize the symmetry, and I used DFT to try to calculate, and the very first number that it pushed out for electronic energy, I would use that number to based on.
Well, I just saw 10 to the 6 kilojoules, and I'm like, that
So-
seems
So yes, I use the auto optimization function on chem compute.
huh.
So yeah,
Cool. Just curious.
So as long as we're on this slide, right? So it seems like one thing that might rule out the bottom path might be the stereochemistry at the bottom left of the molecule, right? So if that O minus is on the opposite face of that 500 ring of that double bond, it won't be able to reach it, so it won't be able to do that.
Oh.
Is there any, and of course that intermediate is that chiral center is made during the reaction. Is there anything that you can think of where you might be able to use it to bias the system stereochemically like that?
that is one really interesting question. Yes, I think there are a lot of potential to that, but like I might need to sit down and dive deeper into that specific one question. But thank you.
Allison again.
Good team. So I'm always impressed by any time y'all make molecules because it's impressive. But was there one particular aspect of the synthesis that was challenging that you got something that you didn't expect, and or there was a particular challenge that you just need to vent a little bit about?
So one thing that I really into during this synthesis is that here, this molecule. So if I start with a furan, there should be a double bond here instead of the dihydrogen. However, if there's a double bond there, I cannot do the Heck reaction because like I cannot coupling freely. Whatever happened on one side of the molecule should happen correctly to the other side. So I have to find a way to get rid of the double bond here at like position number 2 and 3. And that's where the Birch reduction come in. So I have to manipulate the molecule by using an electron withdrawing groups of the furanic. So this is furanic acid. I have to use the electron withdrawing group, a carboxylic acid, to redirect the Birch reduction to be going into the position 2 and 3 there. So because like Birch reaction is using alkaline metal, using sodium and lithium in the liquid ammonia. So basically I will have like solvent electron, so single electron coming into that, like E minus only, nothing else. Poof.
I think that's a really interesting challenge. I think Dr. Fleshman was asking about the work you actually did in the lab.
Oh, actually did in the lab. Okay, so what I actually did in the lab is this particular reaction. Well, the challenge is this reaction here on the paper is supposed to work. However, no matter how many times I tried to recreate it in the lab, I could not get to this 1,2,4 triazole. And yeah, what I tried to go over with this 1,2,4 triazole is that I tried to make the 1,3,4 oxadiazole instead. So going a slightly different route, instead of like 3 nitrogen here, I tried to make like 2 nitrogen and 1 oxygen. So yeah, I tried to detour it a little bit.
I have a question for you. So one thing you didn't talk about in your like malaria save the world pitch was the cost. So it's simpler and the higher yield to make some of these products. Is it cheaper than the extraction process from the plant?
So total synthesis, one fascinating about total synthesis is that total synthesis can always be getting better and better. So you can tinker the total synthesis to eventually reduce the cost. For now, for this particular total synthesis that in the 2012 paper, so it is really high cost comparing to just using extraction because they use a lot of rare metal. They use molybdenum, they use palladium, they use like expensive catalyst to make that molecule. So it only serves the total synthesis purpose, like making the molecule, but not the economically way yet. But in the future, eventually, we can get there to the point that we have like really high yield and we start optimizing for reducing the cost.
That's good news. Good news for us. Other questions for our speaker? If not, let's thank Chris again. Join us on Friday for our final 2 senior seminars, 3:10 and 4 o'clock. Thank you.